So we have seen the necessity of reproduction and we learned about the variations. We learned that organisms reproduce and produce the young ones of their own kind. Every organism produces the young ones of its own kind, which is similar to that but not exactly identical. Now we are going to discuss how the organisms produce the young ones of their own kind. By what method? Do all organisms follow a similar method of reproduction? No. Different organisms have different methods of reproduction depending upon their complexity of their body, complexity of their organization. There are various methods of reproduction. So organisms propagate in different ways, different methods. Let us see the modes of reproduction used by single-celled organisms. What kind of reproduction is observed in single-celled organisms? The organisms that have only one cell throughout their life are called single-celled, or we can call them as unicellular organisms. So here, if you see this, the modes of reproduction used by single-celled organism, the major one is fission. Fission. It is an asexual mode of reproduction, means that is from a single cell, the production of new organisms takes place. It is of two types. The fission is of two types. Binary fission the second one is multiple fission. The name itself tells us that binary, one cell, it divides into two daughter cells, that is binary fission. Multiple, one cell divides into many daughter cells. This is called multiple fission. Here cell division takes place. It is at the cellular level. That means the cell it divides. Very simple. This is the very simplest form of reproduction. Here in binary fission, you see the bacteria is an example for binary fission. If this is the cell of the bacterial cell, it consists of some genetic material, that is the DNA present inside its body without any cover, without any nucleus. Genetic material is directly present in the cytoplasm. So what happens here? The wall of the bacteria, it takes a constriction like this, folding. Now the genetic material also starts divide into two. Here the genetic material, here the genetic material. Now this wall, it grows up, and finally it attaches like this, and both the cells get detached. Now these are the two daughter cells, separate two daughter cells that are formed from the parent cell. So now there is no question of parent. Where is the parent? So here the parent has become into two cells. Now you cannot tell which is parent, which is daughter. Parent is not existed here because the parent cell is divided into two daughter cells. You cannot say this is parent, this is daughter. We cannot distinguish it like that. This is the binary fission in which a cell splits into two. A cell divides into two. First the genetic material, it is divided, then the cytoplasm is divided, and whatever the cell membrane is there, the cell membrane, it constricts like this and divides both the cells, and they are formed as two individual organisms, daughter cells. So this kind of binary fission is observed in organisms like bacteria, and this is also observed in protozoa. Protozoans, we observe this binary fission. We can say, take the example like amoeba. In case of amoeba also, it splits into two. Amoeba binary fission we see, paramecium, euglena. And one more important point that we have to notice here is that in some of the organisms, this binary fission, it follows a specific orientation. That means, for example, here is an organism called amoeba. If the amoeba splits at any orientation, any axis, the two daughter cells you can easily form because the amoeba is not having any specific shape. But if some organisms you see, they will be having some special structures. See, for example, Leishmania, it is having a whip-like structure. A whip-like structure. So the division takes place at particular orientation, at particular axis. Even the binary fission in case of paramecium, it happens at a particular orientation, particular direction. So that is observed in binary fission. Anyway, binary fission means the division of the organism into two daughter cells, two individual daughter cells is called as binary fission. This is a very simple mode of reproduction which involves only one cell, one organism is involved. So this is binary fission. Whereas if you see the multiple fission, here one organism is divided into number of organisms. One parent cell divides into number of daughter cells. Here we take the example Entamoeba histolytica, Entamoeba. So this Entamoeba, it reproduces by multiple fission. So here the Entamoeba histolytica, this is the case in which one single organism is divided into a number of daughter cells. This is called as multiple fission, means one cell, one parent cell to many daughter cells. So this is a method of reproduction that is used by single-celled organisms. So the mode of reproduction in simple organisms, that is single-celled organisms, and even though the multicellular organisms, if you see certain organisms are very small, that means they consist of very few number of cells. And even though all the cells are of same kind, for example, if you take Spirogyra, it is a multicellular organism, but the cells are similar. But if you see a big animal, if you take human being, we are multicellular, even then in our body all the cells are not alike. We are made up of various kinds of system. Each system is made up of various organs are made up of various tissues. Each tissue consists of a specialized cells. That means all cells are not of same type. So cell by cell division is not possible in higher animals because the cells are differentiated into various tissues, different kinds of tissues. So differentiation has taken place in complex organisms. But whereas in the simple forms, that is in case of the single celled organisms or in case of the multicellular organisms which are very simple, we can find some modes of reproduction which are very simple. So we have seen the fission. It's a very simple mode of reproduction in which a cell divides into either two or many. If it is two, you call it as binary fission. And we learned the examples like amoeba, bacteria, and certain protozoans like paramecium, etc. Paramecium, euglena, leishmania, whatever. So we have discussed it here. Multiple fission examples: Entamoeba, and Plasmodium. Plasmodium is also a protozoan. Plasmodium is also a protozoan. The Plasmodium, it causes malaria in human beings. This Plasmodium also reproduces by multiple fission method, Plasmodium. So these are the examples of multiple fission. These are the mode of reproduction in single-celled organisms. Now let us see the budding. Budding is found in multicellular as well as unicellular. In case of unicellular organisms, if you see the yeast is a kind of fungus, but it is unicellular. In fungus there are so many species, but yeast is the only species which is unicellular. Other fungi are multicellular. So yeast is a unicellular organism. How it reproduces? Yeast reproduces by a process called budding. This is a mature or grown up yeast cell. When the yeast is completely mature, it produces a bud on its body. On its body it produces a small bud. So this bud, it grows up again, it produces a bud. It may produce a bud. It may produce many buds. This way the budding takes place. So these buds, they get detached from the mother cell, the parent cell, and they grow individually into new yeast organisms. So this is the way they reproduce. This is also the simplest mode of reproduction seen in single-celled organisms. However, in case of multicellular organisms also budding is seen. We will discuss it later. The next one is fragmentation. Fragmentation is the simplest method of reproduction in multicellular organism. Example, we can take Spirogyra. So the Spirogyra, whenever it is mature, whenever it is completely grown up, the Spirogyra filaments, you might have seen the Spirogyra filaments in your laboratory on the permanent slide. It is a green color. Spirogyra. All cells are similar. So it splits into many parts. Now each individual part can grow into an individual Spirogyra. This is called as fragmentation, becoming fragments. Each fragment can grow into a new organism. But can we apply the same concept how the Spirogyra, whenever it is split into different parts, each part can grow into a new organism? Can we apply this mode of reproduction to higher organisms? Why can't humans reproduce like that? It is not possible. Because I already told you, in higher organisms, all the cells are not alike. In our body, the cells are differentiated into various systems, and each system is comprised of various tissues. That means all the cells are not alike in our body. So differentiation has taken place. But whereas in the simple form, in Spirogyra, all the cells are alike, so there is a possibility of fragmentation. They can use this fragmentation as a mode of reproduction, but that is not possible in the higher organisms. So they have other means, they have other complex means of reproduction. So now, after fragmentation, we can see one more method, that is regeneration. So the next one is regeneration. What is this regeneration? Here, this is a method which is observed in many multicellular organisms which have specialized tissues. In the earlier case, Spirogyra fragmentation, it is not having any specialized tissue, same kind of cells throughout its body. If it is cut into many parts, then each part can be grown into individual organism. That is fragmentation. Here we see regeneration. What is this regeneration? Here the best examples we can study under this regeneration are Hydra and Planaria. If these organisms are cut into different parts, if a Planaria is cut into different parts, each part can grow into an individual organism. This is regeneration. This is observed in many organisms. That means, so these organisms, they have the ability, here this part is cut. How can this part produce the complete organism? Whatever the part that is cut, it proliferates. That means cell divisions takes place and it forms a mass of cells. And these mass of cells, they differentiate again into the organism. That means initially all the cells are alike, but slowly they develop into different organs, different kinds of cells. This is differentiation. Certain cells in the body of organisms, they can differentiate. Even in human bodies also, in case of humans also, certain cells have the capacity of differentiation. Say for example, there is a human. So just to you compare this fragmentation in the Hydra and Planaria. If the hand of a human is cut and it is kept separate, can the new human come from this hand? Is there any possibility? Just like in case of Hydra or Planaria, in case of Planaria. In case of Planaria, if you cut it into small pieces, each piece can grow into an individual Planaria. Whatever the cut part is there, that part can grow into a group of cells. Now this group of cells differentiate into complete organism. So that is possible. But in case of multicellular organisms, this is in multicellular. Why it is not possible in humans? In humans it is not possible. But in our body also there are some cells which can differentiate and produce an organism or produce whatever the part we like. Say recently you might have heard about stem cell banks. In our bodies, we have some specialized cells called as stem cells. These cells are more in number when we were born. Especially these cells were found in the umbilical cord. That is, the newborn babies you see, the newborn baby will have a cord attached to the mother called as umbilical cord. So, whenever the baby is born, this umbilical cord is cut. Now, the stem cell banks, they are preserving the part of this umbilical cord with stem cells. They are preserving it for future use. Why they are preserving it? So, if anybody has got their baby, so immediately the baby is born, they can keep the stem cells in the bank for their future medical uses. How? For example, there is a person called as X and Y. So, father X, mother Y. They have a baby called as Z. So, when this Z is born, his umbilical cord is preserved. When there is a problem with X liver, father's liver got damaged, there is a problem. So, father needs some liver. A part of liver has to be transplanted. In such cases, whatever the umbilical cord that is stored from this baby in the stem cell bank, from that umbilical cord, from that stem cells, the organ can be developed and kept in this person to save him. So, in this way, in humans also, the doctors and scientists are finding the possibilities of differentiation, generating the complete body or at least some parts of our body from some cells by different methods. So, here in regeneration, we can understand this concept, Hydra and Planaria. These organisms, if they are cut into different parts, each part can differentiate into a complete organism. That is possible. But humans, it is not directly possible, but in case of stem cells, it is possible. By preserving our stem cells, we can get our body parts in future, artificially developed in the labs by using our stem cells. So, that is the case in case of regeneration. So, budding is another method of reproduction, another mode of reproduction in which a single organism is involved. Budding already we discussed in unicellular organisms, that is yeast. So, budding is seen in unicellular organism, that is yeast, and in multicellular organism, that is Hydra. So, Hydra is having tentacles and the body like this. So, this Hydra, it produces a small bud on its body, surface of its body. This bud gradually grows up and it detaches from the parent and it grows as an individual or separate organism. So, this is what called as budding in Hydra.